Okay, did everybody get a little handout from last time? Yeah, up there by the exams. Okay, let's see. And it is on Moodle too. How you doing?
A lot better. Everyone in my house has it though. Well, when I started watching the video at first, like I fell asleep halfway through.
Oh yeah. Yeah. No. That's a yeah.
And in the fall. There's only really 2 super bad days. Every other day was like a bad cold. And the one was really weak.
Oh yeah. Yeah. All right, let's see. I'm gonna try and share content. Sorry, Bart. I'm not running on time. Well, I'm not too far off. Okay. Here we go. Did we not get through all the organs last time? They're only numbered 1, 2, 3. Okay. I was like, did we really only do that? But I guess we got caught. Exams up here. Okay, so what do you tell your fruit when you're leaving the house? I'm going bananas. Yeah. It's cute. Okay. Oh, this one's gotta go. All right. So exams are back. So I'll give y'all a minute to kind of look over your exams. You also need the little handout. Sam, it's on Moodle. It's 4 questions. All right, 4 questions. Yeah, chapter 19, metabolic integration. Everybody got one of these? Yes, chapter 19 handout. Yes, Sam. Giving everybody just a minute to look at their exam. Y'all did pretty well. I do feel like I go through like a red ink pen for every exam.
Maybe you should
Just not.
let some slide. Save the world. Use less.
Use less ink. I do feel like even if I don't take off a lot of points, I feel the need to fill in gaps for y'all and, you know, that kind of stuff. Yeah, that's fine, Sam. Whenever. See if you can start working on, if you're done looking at your exam, see if you can start working on the other, we're going to pick up on 4. We've got adipose tissue, we've got muscle, and kidneys left. No, I didn't do it. That would be great, wouldn't it? But now my clicker was acting up on this computer the other day, where it didn't want to. It didn't want to let me sign in. Let's see. See, I don't understand. It says it's in use by another base, but it's not. Y'all can try. I don't know if it's gonna work. Oh, it did. Didn't work for me yesterday, which is hilarious. So it's working. Cool beans. I'm gonna make this full screen. Okay. So y'all ready? So exams are in the front if you want your exam. Take a look at it, but turn it back in before you leave. And I have a meeting like right after class. Like I have to run out of here today. Sorry, it happens. It happens. Okay. So what we're talking about is flux in the vital organs, right? So what are the primary functions that are required to maintain metabolic homeostasis, right? So we talked about number one was the liver, right? Always, of course, number one's the liver. Number two was the brain. Number three was heart, right? The heart was kind of special. Okay, 4, adipose tissue. So tell me about adipose tissue. What's its job? Energy storage, but now we're not talking about just storage, we're talking about metabolic flux. So if it stores fatty acids, what does it do with its stored fatty acids? Does it just keep them and never shares them? No, it will share. It will share. So it'll convert it, right? It'll exchange fatty acids and triacylglycerols, right? Between what two places, basically? We talked about this in one of the case studies. Liver. So between liver and adipose. So you have that maintenance of fats inside of your circulation, right? So maintain fatty acids in circulation. Right? If they're in the blood, they're not free fatty acids, they're not triacyl. Well, they can be triacylglycerides, but they can't be fatty acids. So if they're in the bloodstream, in what two forms are they in? You can't have free fatty acids in the bloodstream, right? That's like snake venom. Not a good thing, right? So what are they bound to? Albumin. Another way that you can transport them in the bloodstream is in give me something that's kind of like all encompassing. Yeah, lipoprotein. We'll put ETC, right? Dot, dot, dot. Depends on what form they're in. Okay. All right. What is 5? 5 is our skeletal muscle. Skeletal muscle. Okay, so what is our skeletal muscle going to do? Is it going to give off anything? No, it's a very, very, very selfish tissue type, right? Selfish tissue type. So what is it going to do? It's going to use its glucose. And it can also use fatty acids, right? Depends on whatever it has. If it runs out of the glucose, then it's going to convert to fatty acids, right? From, we'll say, diet. And depending on if we get it from the liver, for energy, okay? Then what happens if we're anaerobic? We go through glycolysis. Can we go through oxidative phosphorylation? No. So what product are we going to make from glycolysis? Well, we're going to make pyruvate, and then we're going to go through fermentation, and we're going to make lactate or lactic acid, right? So you could say export lactate, lactate, right? And what cycle is it that it sends the lactate to the liver? Liver will convert that back through gluconeogenesis. What is it? Cori cycle. So I'm just jotting things down. Y'all should tell me like, hey, Dr. B, we can't see the screen. I'm just jotting things down, right? So this would not be a good discussion answer. This is just me jotting things down, right? Y'all understand that? I mean, like if I ask you, what is the role of skeletal muscle in maintaining metabolism in the body? You couldn't just jot these things out. You have to explain them. Right. But this is just for us to kind of get all of our thoughts together, right? Okay. All right. So 6. What's 6? Kidneys. That's the last big one. Okay. So what's the job of the kidneys? Right, to filter blood, absolutely. And to do that, it's going to have to expend energy, right? It's going to do all the normal cellular processes and stuff. So it's going to have to break down amino acids just like everybody else, right? So what's it going to do with that excess nitrogen? So excess, we'll say NH4 plus, right? From protein degradation. Urea happens in the liver, DGREG degradation, to liver via what? Certain amino acids, 2 certain amino acids. Close. One of them is glutamine. The other one is an A, alanine. And we'll just say for excretion as urea. Right? So these are all of the main vital organs that require a metabolic homeostasis role. Okay, pretty good. Kind of put them all together, kind of think about them all. All right, so I want to take a step back and go back to our fats, our triacylglycerols. So remember how we said we're going to convert and move fatty acids from the adipose tissue to the liver, from the liver to the adipose tissue, and back and forth and back and forth. And the whole point of doing that was to maintain a certain level of high energy compounds in the bloodstream for our cells to use, right? So we need to kind of talk about that, how that's connected, right? So honestly, about 75% of the fatty acids that are released from adipose tissue come back as tags. Fatty acids released from adipose, AD, adipose, come back and are remade into triacylglycerols. So how much of it gets used? Not very much of it, right? Not unless you're under starvation conditions or something like that, right? But you have that stockpile of high energy compounds that are not only stored in an organ and inaccessible, but in circulation, ready to be used. So remember remember the little patient who had our glycogen disorder? Do you remember that, right? And he would run through his glucose very quickly, couldn't use glycogen, and so he would switch to an alternate fuel source, right? Was the downtime for his second wind really long? No. So it doesn't take long because the high energy compounds are continuously around in the bloodstream so that they can get to it really, really quickly. So what do you need to have in order to maintain this cycle, right? What are the main 2 components of a triacylglycerol? You need a glycerol and fatty acids, okay? So The fatty acids is where the storage for the energy is, right? And the glycerol is the backbone. So really, if you have enough of the glycerol, you can do this cycling, right? So you need to have a backbone, and the backbone, that substrate that we have to have is glycerol 3-phosphate. So when we talk about glycerol 3-phosphate, you have to talk about it in two different ways. Yes, there is a systemic circulation. So when we talk about systemic circulation, this is where we're talking about converting from one organ to another organ. So we're going from adipose to the liver, right? So this is where we're doing that free fatty acid combined with albumin, ALB, and our lipoproteins. Right? Now, intracellular. When do we have to circulate between triacylglycerols and free fatty acids intracellularly? This is our adipocytes. Why can't I spell adipocytes correctly the first time I write it? I just can't, right? But this is that cycle between the free fatty acids, right? Because you have that fatty acid binding protein in the cytosol. Remember that? So it cycles between the free fatty acids and the triacylglycerols in the droplets, right? The lipases will cleave it to get free fatty acids from the triacylglycerols, right? And back and forth. Okay, so when we talk about the availability of glycerol 3-phosphate, we're talking about really, you need glycerol 3-phosphate in two different places. You need it in adipose tissue and you need it in the liver, right? So how can you get glycerol 3-phosphate? The easiest way to get glycerol 3-phosphate is to take glycerol and what? Phosphorylate it. You could do that in the liver. Guess what? Adipose tissue has no glycerol kinase. So can you just phosphorylate it in the adipose tissue? No. So what are you going to do? How are you going to generate it? You can generate glycerol 3-phosphate from dietary glucose. So in dietary glucose, how are we going to get glycerol? We're going to have to start breaking down glucose. What pathway do we start with? Glycolysis. Do you remember when you cleave the glucose molecule in half? What are the two things that you get? One of them is DHAP, right? Dihydroxyacetone phosphate. You remember that? Yes, maybe slightly. So we can use glycerol 3-phosphate dehydrogenase to convert dihydroxyacetone phosphate to glycerol 3-phosphate. So we can get our backbone core that we need in order to make triacylglycerols, right? So here, what are we doing with this? If we're in the triacylglycerol, this is being used, stored. If it's in adipose tissue and it's in the triacylglycerol stage, storage or use? Storage. Okay. Now we want to make sure that we have enough in the bloodstream, so we're going to hydrolyze some of those triacylglycerols with our hormone sensitive lipases, right? Which means that we're going to cleave the free fatty acids from the glycerol. What happens to the glycerol? It shipped out to bloodstream, goes to the liver where it could be used to phosphorylate to get glycerol 3-phosphate, right? But why do we need glycerol 3-phosphate in the liver? Well, we've made free fatty acids, so we can take those free fatty acids, we can put them into circulation into the bloodstream, right? So some of the peripheral tissues will use those fatty acids for energy, right? So here we're going to use for energy. But what percent was that? 25%, right? Only 25%. what's going to happen to the rest of it? The other 75%, right, is going to go back into the liver, but we can't have free fatty acids, right? That's not going to work. So the liver is going to convert it back into triacylglycerol because it now has glycerol triphosphate, which it can use to make triacylglycerols. Now that we have triacylglycerols in the liver, well, we can ship some of those out to peripheral tissues, and this also contributes to that 25%, right? So these two contribute to the 25%, right? So we have those triacylglycerols. What is this LPL? Lipoprotein lipase. We can cleave some of them to make glycerol and fatty acids, right? Then what happens to it? Sorry, I gotta go this way. Attach it to glycerol 3-phosphate, we can store it again. It's kind of a complicated little mechanism there. Yeah, but you're going to have constant cycling. Okay, so what happens if we're low on glucose? Where do we have an issue if we're low on glucose? Making the dihydroxyacetone phosphate. Here's where I have an issue, right? Because how am I going to make glycerol if I said that I'm going to get it from dietary glucose? If you're low on glucose, so when blood glucose levels are low, the DHAP is synthesized from amino acids and lactate, right? Because we can get pyruvate. If we can get pyruvate, then we go through what is called Glyceroniogenesis, creation of glycerol, right? So some of the enzymes that are in the pathway are glucagonogenic enzymes, and some are just the reverse of glycolysis. So it's kind of like a mix and match of different enzymes, right? So the glycerol 3-phosphate that we make, right, is only used for glycerol 3-phosphate synthesis, right? So it's a very specific pathway that substrate is going to be used for, right? And why is that? Why are you not going to use it for gluconeogenesis? What cell type are you in? Fats. Do fats make sugar? No. Why don't fats make sugar? Fat's job is to store energy in the form of fat. So you don't want it to be making sugar when it should be making fat. So how do you prevent that from happening? Adipose cells do not express the enzymes that are required in gluconeogenesis. That's how we make sure that this substrate is not used for gluconeogenesis. We don't have the enzymes for it, right? That's how most of these mechanisms are controlled in different cell types. You don't have the right enzymes to do whatever process that you shouldn't be doing. It's pretty cool. All right, so in glyceroniogenesis, I just want to, before we look at this like super complicated pathway, I just want to give you an idea of what's going to happen, right? We're starting with pyruvate, right? And pyruvate can come from, what did we say? Pyruvate can come from degradation of amino acids and lactate. Okay. So we have pyruvate. We're going to convert it to phosphoenol pyruvate. What is that a substrate in? Glycolysis, gluconeogenesis, right? To dihydroxyacetone phosphate, to glycerol 3-phosphate. So what's interesting is this requires both mitochondrial and cytosolic enzymes, right? So whether you're coming from amino acids or lactate, whatever, doesn't matter, you're going to make pyruvate, right? Pyruvate can enter mitochondria. Once we have pyruvate in our mitochondria, they can go through and be converted into oxaloacetate. Do you remember this was another way? It's pyruvate carboxylase. So this was another way to make more oxaloacetate. Do you remember when we were talking about regulation of citrate cycle way back when? So it's just another way to make oxaloacetate. Now oxaloacetate can either go through half of the citrate cycle to make malate, or you can just go straight through malate dehydrogenase to make malate. Now malate can be shipped out. Malate dehydrogenase, oh, we went back to oxaloacetate, but the point is that oxaloacetate is now in the cytosol. Whereas if it's in the mitochondria, I can't do anything with it because the other enzymes are what? Glucagonogenic enzymes, which are found in the cytosol.
Why does it go through half of the citrate cycle with oxaloacetate? It's just made into malate in one step.
It goes through. Well, so why do you go through either way?
Oxaloacetate, so it's reversed.
Yeah,
So it goes like backwards.
yeah, but it can go either way. All right, so now we have oxaloacetate and we have it in the cytosol, right? So now that we have it in the cytosol, we can go glucagonogenic reactions, right? We're gonna have phosphoenol pyruvate carboxykinase, and then we go through our glucagonogenic reactions. We make phosphoenol pyruvate, go backwards, right? DHAP, 2-glycerol 3-phosphate, right? So some things have to be done in the mitochondria, some things have to be done in the cytosol. Yeah? All right, now, good on this? Maybe? All right, on your sheet in front of you, right? You have, I don't have a sheet. On this sheet right here, this little handout, right? For 4 little questions. We're gonna start tackling 1 and 2. Describe in detail the primary response of insulin and glucagon signaling in the liver, skeletal muscle, adipose tissue, and the brain. That looks like a really small question, doesn't it? It's gonna get real complicated. So, what maintains metabolic homeostasis? Hormones, right? Hormones. And hormones initiate signal transduction cascades. Yeah? Our 2 big hormones, look at number 1, are what 2 hormones? Insulin and glucagon. Insulin is mimicking our fed state, right? We only secrete high levels of insulin when we're fed. And then our hungry state is glucagon. So generally, what happens, right? If we're secreting insulin, regardless of whichever hormone it is, the hormones come from the pancreas. The pancreas has a lot of different cell types, and each of the different cell types makes a different hormone. So the beta cells secrete insulin, the alpha cells secrete glucagon, and we'll talk about them in a little bit more detail. So if I secrete insulin, right? What do I want that insulin to do to basically general body cells? Take glucose in, right? Take glucose in, phosphorylate it, use it for an energy source, right? That's liver, that's skeletal muscle, that's adipose tissue, whatever, doesn't matter. Okay, now what if I am skeletal muscle or liver? There's something special that those 2 cell types can do. If I eat enough sugar, right, and my insulin's high enough, and I don't need any more energy for cellular jobs, I'm gonna make glycogen. What else can I make as a storage molecule? Fat, right? So anything excess, my body is preparing, winter is coming, right? Winter is coming, so I need to store. I have enough, right? So I need to tell my body, stop eating. So you stop feeling hungry. So you're actually also going to affect neurotransmitters in the brain to make you stop feeling hungry. right? You'll get that satiated feeling. Glucagon's the opposite. Glucagon is also produced in the pancreas, but in a different cell type. That's the alpha cells. So if you're secreting glucagon, how long's it been since you've eaten? A couple hours, right? A couple hours, getting hungry. You know, you're sleeping, whatever it is. So you release glucagon from our alpha cells. And what's that going to tell the body to do? There's not enough energy, right? The brain needs glucose in order to work. So what do we need to put into the bloodstream? Glucose. How are we going to put glucose into the bloodstream? A couple of different ways, right? We could make it through gluconeogenesis. We could degrade glycogen. We could also break down fatty acids, right? So those are our three different ways that we can handle that situation. So now what we're going to do is we're going to look at these two hormones and what happens in the pancreas, and then we're going to take this out to the two other cell types. All right, so here's our pancreas, right? Our pancreas is attached to the small intestine, kind of like the very beginning of the small intestine, the duodenum or duodenum. It depends on who you are, how you pronounce it, right? I always heard duodenum, but then I had a friend who was taught duodenum. So then we make fun of each other for what we say. All right, so let's talk about the different cell types that are in the pancreas. In the pancreas, you have an area, right, that's called the Islets of Langerhans. And so it has a lot of different cell types inside of that one specific area. So let's kind of zoom in. So you have alpha cells. Alpha cells are specifically going to make glucagon, right? You have beta cells. Beta cells are going to make insulin. You have delta cells. Delta cells make somatostatin. So somatostatin is a local hormone, right? What do we call a local hormone? Paracrine, one that will work on cells that are in the immediate area, right? So as you eat, you're going to change your somatostatin expression. Your somatostatin expression is going to regulate insulin, glucagon, and your digestive proteases. Right? So that's like your local hormone that controls all the different things that happen inside of your pancreas. Right? Okay. So you're not actually going to secrete the finished hormone. For example, like insulin is secreted, it's made, and it actually has more amino acids than it needs, and it actually has to be cut in order to be functional, right? So both glucagon and insulin are like that, but we won't talk a whole lot about that, right? But okay, let's talk about this real quick. If I have high blood sugar, high BS, right? High blood sugar. What's going to happen in the pancreas? The first thing when glucose enters the cell, what's going to happen to the glucose? It's phosphorylated. What enzyme is going to do that phosphorylation? Be careful. Remember back in Biochem 1, I told you that there were two cell types, glucokinase. So we stimulate glucokinase to phosphorylate glucose, right? This is why the pancreas is that glucose sensor, right?
Is this after insulin brings it in?
No. This is what stimulates insulin. This is what stimulates insulin. So the pancreas is what responds to the high levels of blood sugar. You eat, right? The foods that you eat are going to be broken down. They are going to result in high blood sugar. Insulin doesn't cause high blood sugar. High blood sugar results in insulin production, right? So how does that work? Well, if you have high blood sugar, you stimulate glucokinase. Glucokinase phosphorylates the glucose. If the glucose is phosphorylated, that increase inside the cell of the products of glycolysis are eventually going to do that signaling cascade. You can go back to, what chapter was that? 9, way back when, right? If you want to see that pathway again, eventually to release insulin, right?
That's what brings the phosphorylated glucose
Into other cell types, right? Into other cell types. And we're going to talk about why it's different.
How is phosphorylated inside the cell travel?
It is. It is.
But it's transported as it's phosphorylated before getting trapped in.
Wait, no. So you have blood, you have sugar in your blood, right? So it's all over your blood, all over your body. Your body cells are not taking it in. Your pancreatic cells are taking it in. They have a different transporter. So they pull it in, they phosphorylate it. That induces the release of insulin. Then once insulin is also in the bloodstream, in addition to having high blood sugar, now your body, your other body cells are going to be signaled, hey, you need to turn on the receptor, the transporter, really not the receptor, but the transporter that will pull glucose in and allow you to phosphorylate it and keep it and degrade it. That answer your question? Okay. So the release of glucagon is way more complicated, and so we're not going to talk about that. We're not going to talk about that, but you're always going to release both, actually a little bit of both insulin and glucagon, but the ratios are going to be different depending on if you have high blood sugar or low blood sugar. But we're really going to focus a lot more on insulin than we are on any other cell type. Okay, so this is a really nice figure because it looks at our 4 major tissue types and compares what happens with insulin signaling versus glucagon signaling. So what's really important to notice right off the bat is in insulin signaling, all 4 cell types that we have listed here, liver, skeletal muscle, adipose, and brain, these all have receptors that will increase expression of a glucose transporter, right? Glucagon signaling only occurs in liver and adipose tissue. Skeletal muscle has no glucagon receptor. Brain has no glucagon receptor. What are your selfish organs? Brain and skeletal muscle. So do they need to be responsive to glucagon? Absolutely not, right? Do you ever want your brain exporting sugar? No, it needs sugar. How about skeletal muscle? You ever want that giving up its energy? No, that would make no sense, right? Okay, so you have 4 targets for insulin, 2 targets for glucagon. So when you're looking at this question number 1, right? This is actually what we're answering right here, number 1. For 2 of the tissues, you're not going to have an answer, right? You're just going to say no effect. So let's kind of look through insulin signaling, and what we're going to do is we're going to go through all of the pathways. Okay. So one thing I do want to say in this table, they give you, it stimulates glucose uptake by increasing the level of GLUT4. What is GLUT4? It's a transport protein. So there are different transport proteins depending on the cell type. Some of them are responsive to insulin levels, blood sugar levels, and some of them are continuously, it doesn't matter if the blood sugar goes up or down, the same number of transport molecules are there. So if you see GLUT4 is one that is sensitive to insulin signaling. Okay, so let's talk about the organ. So this is kind of what I want you to do for each of the organs. So number 1, let's start in the middle. I'm telling you that we're secreting insulin. Why are we secreting insulin? Start in the middle. Okay, so we have high blood sugar. High blood sugar. So that tells us that what's happening. That we just ate, right? So we ate, so we have high blood sugar. We have high blood sugar. This activates what? Pancreas to release insulin, right? That's where our insulin is coming from. So I guess I should kind of do it that way instead of, why did I do that? Okay. All right. So now what I want to do is I want to break down everything that's here. What happens when liver is exposed to insulin? Okay. Protein phosphatase 1. Let's talk about protein phosphatase 1. Do you remember that was a signaling molecule in that massive cascade mechanism that we talked about way back when? One of the things that protein phosphatase 1 does is that it activates phosphofructokinase 1. What does phosphofructokinase 1 do? It what? Rate limiting in glycolysis. So if we promote it, what happens to glycolysis? You're going to increase activity of glycolysis, right? Okay. I'm also going to stimulate glycogen synthase. What's that going to do? We're going to make glycogen, right? So for every one of these, I'm kind of analyzing it and trying to figure out what each individual step does, and then what's the overall effect on the tissue. Okay, phosphorylase kinase and glycogen phosphorylase. Phosphorylase kinase works on an enzyme that is required for glycogen degradation, right? Remember that? Sort of, maybe. Now do y'all know what to go back and study? Are you seeing this? So these two are important to inhibit glycogen degradation. So I'm going to inhibit both phosphorylase kinase and glycogen phosphorylase. And so I inhibit glycogen degradation. How about this one? Fructose 1,6-bisphosphatase. Do you remember that one? That was a little complicated. That's in chapter 14. Chapter 14, if you want to go back in gluconeogenesis, so remember, you'll have to go back and review. I mean, like, we could spend a whole day going back on that, but you're going to decrease your activity in gluconeogenesis. So far right now, we've decreased gluconeogenesis, increased glycolysis. We have stimulated glycogen synthesis. We have inhibited glycogen degradation. What are you seeing? Opposing pathways. When one's up, the other's down, and vice versa. So you're always going to see that. Okay. Glucokinase. Glucokinase is going to do what? Phosphorylate glucose, trap glucose in the cell, right? Okay, Pyruvate dehydrogenase, protein phosphatase 2A, acetyl-CoA carboxylase. What are these all important in? Fatty acid synthesis, right? So why do we want to increase fatty acid synthesis? Same reason why we want to make glycogen, right? So if we have enough glucose, we're going to stimulate these storage pathways. Now, is the liver sensitive to how much glucose is coming in? Is that regulated by insulin? No, because it uses the GLUT2 transporter, which does not change expression levels. So it doesn't matter how much is there. It's not going to change the concentration of transporters on the outside of the liver.
There's always like a high expression of glucose.
Yeah, it's like always on the outside of the cells.
So like
So glucose can come in and can come in regardless.
right. So like the ones that you put before that
Like the
expression
muscle.
changes.
It does. It does. Okay. I have done one for you. How many you got left? 3. I want y'all to work on that right now. But do y'all see, do y'all understand you're looking for big picture effects and you're looking for individual things. So y'all can work together, but I want you to break down each of them.
Dr. B, on the final, it's gonna be, we're gonna be asked questions from every chapter, or just like-
Some questions are going to be like, more questions are going to be integration. You know, like
So more like 19.
more like 19. It's going to be heavy 19 because it pulls in all the
We still
pieces.
should go back over.
So what should you go back over from here?
Black fatty acid synthesis, glycolysis, glycogen synthesis, glycogen degradation.
This is all.
Right? These are the big
Less
ones.
Like of the super specific from each.
Correct. Less of the super specific from
And
each.
big
More
picture.
big picture integration.
But this is no like everything. Like, yeah.
Yeah. But you actually have been tested on it, but you haven't been tested on it, right? Yeah. Yeah. That's the stuff that's going to be heavy on the exam. That makes sense. Okay. So you got to work together. We'll go over them. But basically what you're doing is you're answering number 1, right? You're answering for the 4 main tissue types, what's insulin's effect? You're answering number 1 right now. What time is it? 9:47. 10 minutes? And if you finish, go on and do glucagon, right? Because the next one is glucagon and there's only 2 tissues, so it's a whole lot less. Okay, this one, this diagram, this is the only thing I want to hear y'all talking about. Oh, yeah, I'm gonna go home with starvation. I don't know if we'll get there. Y'all ready? Okay. So skeletal muscle. You want to do that one? All right. So what happens in skeletal muscle? GLUT4. What does this do? Make more glucose. Okay. So if we intake more glucose, what pathways are we going to stimulate? Now this is muscle. This is skeletal muscle. Skeletal muscle. You're not going to make fats in skeletal muscle. What else does muscle use? Wait, what else does muscle use if you have lots of sugar? Like I'm going to go, this is so not me, but I'm going to go run a marathon. So I'm going to carb load, right? So I'm going to have glucose at the ready in my bloodstream, but what else, where else are those carbs going? Why do I want to carb load? I need to make glycogen. Right? I need to make glycogen. Me and that marathon, yeah, right. Okay. So now what else? What else y'all got? So our pyruvate dehydrogenase complex, this one's going to be important for what? Citrate. Okay. What about protein phosphatase 1? That's that signaling molecule, right? And we're going to have really similar effects for in skeletal muscle and liver. We both have protein phosphatase 1, right? But do we have to worry about all the things that happen in the liver? No, why not? because skeletal muscle is, remember, one of those really, really selfish organs, does not want to share, right? So anything that comes in skeletal muscle is going to be used by skeletal muscle. So the pathways that we need to worry about just have to do with glycogen storage versus glycogen breakdown, right? So phosphofructokinase 1, what are we doing? Increasing glycolysis. Glycogen synthase. Increase glycogen synthesis. Okay. Phosphorylase kinase and glycogen phosphorylase. Phosphorylase kinase, glycogen phosphorylase. We're doing the same thing. What are we doing? Inhibit glycogen degradation. HIB, inhibit glycogen degradation. I'm running out of room. It's too much going on. What's our net effect? Right, we're going to degrade as much sugar as we need to for energy. When we've degraded enough for energy, we're going to store it in the form of glycogen and inhibit the degradation of storage, of the degradation of the glycogen, right? So that's our like net effect. How is skeletal muscle different than liver? There are three key differences. Can I do this? Three key differences between liver and skeletal muscle. Skeletal muscle is more selfish. It is. Okay, so what does that have to do with the metabolism of the cell type? We're not going to do what? We're not going to defosphorylate. That's true. We can't dephosphorylate, right? So you're going to have no glucose export. That's true. What else can't skeletal muscles do? Can't make fatty acids. Right? It's not going to be made into triacylglycerols. It's not going to be sent to the adipose tissue. None of that's going to happen. Okay, what's another one? Those are the can'ts. You can't make glucaneogenesis. That's true. So we'll put that in another can't. Glucose from neogenesis, right? This is difference between skeletal liver and skeletal muscle. So muscle, I see what you're saying. Muscle can't. Muscle is all the can'ts because it's selfish. Tell me about in the beginning, like before all these pathways start, when glucose comes into these cells, how is it different in skeletal muscle versus liver? Before that, like just uptake of glucose, is it the same in liver as it is in skeletal tissue? No, it's not the same. Why is it different? What about the transporter that pulls things in, pulls glucose in? So in which one does it need to be upregulated? If you have GLUT4, GLU4 is normally low. You add insulin, GLUT4 goes to the membrane, and you pull in glucose. That's muscle. What do we say happens in liver? It's GLUT2, and it's a constant concentration all the time at the membrane. So it's unchanging. Right? Liver, glucose, and GLUT2, concentration at membrane does not change. Good. Okay. If I carb load, let's just say I carb load, what is the primary mechanism by which my blood sugar will eventually drop? Is it going to be liver or is it going to be skeletal muscle? Skeletal muscle. Even though the liver has different ways to metabolize the glucose, think about volume-wise in your body. What has more mass, your liver or all of your skeletal muscles? All of your skeletal muscles are going to have a much higher mass, and therefore they are going to be the causes for what drops your blood sugar. Right? Okay. What do we got left? Brain. So the brain's just doing what? Neuronal signaling to make you feel full. Now, the brain is consuming glucose. Don't get me wrong, the brain is consuming glucose, but its role in metabolism, other than as a consumer, because we haven't talked about these as consumers of energy, right? We're just really talking about regulation of metabolism. Y'all understand the difference? Okay. So neuronal signaling to say, I've eaten, I need to focus my energy on metabolism, on breaking it down, or I'm hungry and you need to go find something to eat, right? Those are two differences. So then you're going to modify your behavior, right? Let's just say here, we're going to modify our behavior. We're going to say, oh, I need to stop eating, right? But then I'm also going to modify my biochemical activity in the body, right? I'm going to undergo more catabolism, right? So if we're in the plus, we're going to stop eating. We're going to increase catabolism, right? Digestion, all the digestion processes will be up. All the storage things will be up, right? All of that. Okay. All right. So how about, oh gosh, we got to squeeze adipose tissue in here. All right, adipose tissue. What's going to happen with adipose tissue? We got GLUT4, so we have, not expression, but we have a higher concentration of the transporter, increase glucose transporter at the cell membrane. Okay, then what happens? So increase glucose uptake, then what? We're going to increase fatty acid synthesis. We're going to increase, what else do we have to make triacylglycerols? So we're going to make glycerol synthesis. Okay, so the lipoprotein lipase, what is the lipoprotein lipase for? If we want to stimulate lipoprotein lipase. So where's the TAG coming from then? Why, wait, yeah, how is that different? Right, we're going to increase glucose uptake, increase fatty acid synthesis, increase glycerol synthesis, but why do we need lipoprotein lipase to break things down? Where's the other source of fats? The dietary fats that are circulating, right? They're in our LDLs, they're in our VLDLs, they're in our VLDLs breakdown to promote lipid uptake. Okay, then the last one, I went too far up. Hormone sensitive lipases. Why do we want to inhibit hormone sensitive lipases? right? Well, if we're making triacylglycerols to store, do we want to break them down? No, not really. No, to prevent lipid droplet breakdown. Right, so you'll have less fatty acid export, right? Because that's the end result when we start breaking down those lipid droplets that we're going to export them. Do we want to export them? No, we've just eaten a high sugar meal. Our bodies are, winter's coming, right? We want to store it. Okay. All right, so now glucagon. Change gears a little bit, right? Glucagon, what kind of state? Are we in the fed state? No, we're starving, right? We're hungry. Glucagons, let's do adipose tissue first. So what do we want to do in adipose tissue? We want to export, break down, right? That's our whole thing. So in order to do this, what, if I want to activate protein kinase A, how am I going to activate protein kinase A? Y'all remember way back? Yeah, it's a GPCR, right? And we go through the whole GPCR pathway to get eventually to protein kinase A. So then what are we going to stimulate? We're going to stimulate those enzymes that release our fatty acids from the lipid droplets. So do you remember that? You had to stimulate perilipin. What does perilipin do? If it's phosphorylated, it leaves the droplet, right? And allows the other hormone sensitive lipases to come in and cleave. Remember the monoacyl, the diacyl, the triacyl, right? It cleaves one at a time off of them. So these are all promoting what? Triacylglycerol, right? Breakdown and fatty acid release. Whereas up there, you were trying to do storage. Down here, you're trying to get rid of. All right, ready for the liver? Why isn't skeletal muscle in the brain here? Not since it's glucagon, we don't have the receptors. Okay. All right. So if we have glucagon, right? What does the liver want to do? Let me ask you net result. What do we want from the liver? We want to export glucose. I don't know what I was writing there. We want to export glucose. So what pathways do you think we're going to activate? Glucagoniogenesis. And what else? Glycogen degradation. Right? This is what our goals are for the liver. Yes, Sam. When we're secreting glycogen. All right. So same thing. It's a GPCR, right? So what are we going to activate? Protein kinase A, right? It's the same activated. You go through your adenylate cyclase, right? So you got to say, then you have your cyclic AMP as your second messenger, activate PKA. All right, what are the targets of PKA? Phosphorylase kinase, what does this one do? Correct. Glycogen degradation, good, okay. All right, let's see. What about so do you remember your PFK2 and your fructose 2,6-bis phosphatase? Do you remember that dual functioning enzyme, right? Okay, so for this one, what are we going to end up with reduced levels of? So we have our dual-sided, our dual enzyme, PFK2 and fructose 2,6 bisphosphatase. Remember that guy? Okay, so if we have those, what are we going to result in decreased levels of? Fructose 2,6 bisphosphate, right? You might have to go back and review that one. That one's kind of been a while since we've done that one. So if this part is active, you're going to reduce your levels of fructose 2,6 bisphosphate, excuse me. The ACE will decrease the amount of the fructose 2,6 bisphosphate product because you're going to convert it. So what's your net result if you activate fructose 2,6 bisphosphatase? Activates what? So you're gonna flux through glycolysis. So you're gonna increase glycolysis, right? Because if you promoted fructose PFK2, then we're gonna promote glucagoniogenesis, right? Okay. All right. PFK1. Where we look at PFK1? Phosphofructokinase 1. Right? Okay, let's keep going down the list. Glycogen synthase. Increases glycogen, these two, both of them increase glycogen degradation, right? If you inhibit those two, if you're inhibiting the synthesis, then you're increasing degradation. So you could say either way. You could say that if you want to say that. Decreased glycogen synthesis. Well, I couldn't think of the word synthesis, right? Okay, and then phosphofructokinase 1. What are we doing? Phosphofructokinase 1. So if you want to increase glycolysis, right? That's the whole point of if we have glucagon, right? We haven't eaten in a while. So we want to export glucose to the brain, right? So what we want to do is we want to inhibit the pathways where we're going to use it for energy. We're not going to be using glucose for energy. We're going to be using glucose for storage, right? So we don't want to do we don't want to do glycolysis. We want to do glucaneogenesis. So PFK1, right? We're going to prevent us from degrading it where we want to make it. We want to go through glucaneogenesis. We want to ship it out to the brain. We want to export glucose.
Why do we increase glycolysis at the top where it says synthesis?
If we promote glycogen, no, that should be down. That should be down. That was just mine. Right? We want to export glucose. This is, think about what cell types need glucose. There are two of them. Brain and red blood cells. Red blood cells have no mitochondria, right? So they are 100% dependent upon glucose as a food source. So if we are secreting glucagon, we have not eaten in a while, and we need to feed those two cell types. So we're going to stop using glucose as an energy source. We're going to start making glucose to be shipped out and use a different energy source for the liver. That makes sense? Yeah. So what I didn't get to that I really wanted to get to, and I'm a little bit sad, but it's okay, is that everything we've talked about so far has been hormonal regulation of your metabolism. But that's not the only way your metabolism is regulated. Your metabolism is also regulated by transcription factors that will create long-term changes in your metabolism, right? So we talked about that. And then I also wanted to take y'all through starvation, because I think starvation is really, really interesting on how the body converts from one energy source to another, and how you get energy and how you can live for a very long time without actually consuming food. You can't live for long without water, but you can live for quite a while without food. So but we're not going to get to those two things. Those are two things I really wanted to get to. But I feel like this is plenty. Yeah, it's plenty. So our exam is not until next Tuesday. I couldn't remember if it was Tuesday or Wednesday. Next Tuesday. So y'all at least have a little bit of time to study. The book goes into starvation. The book, like if you're going to med school, if you are going to med school, you're going any pre-professional, you need to work through the rest of the chapter. Like, it's not going to be on the final exam. It's not. But if you want to be successful, it integrates everything we've done so far, and it's going to be really helpful to you. Really helpful.
We have access to the book.
For a certain period of time, and then it gets cut off. But I don't know how long. It depends on which subscription you purchased.
I don't like that.
That's why I don't like online books. I hate online books.
You like
We
physical
take all our
textbooks.
notes and stuff, and now we're gonna lose all of the notes,
Yep,
like the highlights.
yep, everything.
But
Yep.
they don't even have an option to buy the for long term.
There's like 2 days on the iClicker thing that I forgot.
Okay. Not worried.
Okay.
You're good.
Cool.
Don't worry about it. Don't even worry about it.
All right.
What you got, Meredith?
So I was working on the homework for chapter 19 last night.
Yeah.
So this is, I went through it a bunch of times. I'll show you in a second, but it's asking about ATP levels going back to normal after intense exercise. I went through so many options. I'm just gonna say, I just want to double check. It's phosphorylated, right?
The normal levels of ATP are higher. Normal levels are higher.
It's
What does it
phosphor-
say? When levels return to normal,
It's asking whether or not
creatine is normally phosphorylated. Because right, you want to go through your internal. Oh, and I gotta go because I'm meeting.
Yeah, I'm true.
So it is. So it should be unphosphorylated.
Oh, it should be on phosphorylated?
No, it should be. I'm sorry. It should be phosphorylated. So
Okay,
you
because,
consume a lot of energy.
I went through quite a few times. I think it's just the program was acting funky, but this is what I put the first time. It didn't like that.
Does it want lone pairs?
I tried doing like positive and negative on it, but I don't it didn't like any of that. So I could try a lone pair.
Try lone pairs. See if it likes lone pairs. You have unlimited.
Unlimited tries.
Yeah. Try lone pairs. See if it wants the lone pairs.
Also I'm not
And send me an email. It's like, Dr. B, would you check this structure for me?
okay. And I do want to just one more question. Okay.
Sure.
So for the homework and case studies
Yeah.
and all that, I know it's supposed to like round up to
Come on.
start.
Y'all can come in. Y'all can come in.
Yeah, we're done. Supposed to round up to
Yeah.
the values at the end, but it looks like it rounded up slightly weirdly. I'm supposed to be at 78, but it says 77 right now. So are you just gonna like look that over at the end and like do the calculations and stuff? So I have full points on everything. On the case study, on the what
Sodium carbonate.
The homework.
case study, homework,
And
and
reading
reading
quizzes.
quizzes. So yeah, full points on that.
But this is saying 98%. It's not full.
It says.
Sodium carbonate.
So should I be going off the percentage or the points?
The percentage. I don't understand.
the sodium carbonate acts like a base.
Oh, okay.
And it attacks the,
Go off of the percentage, so this is
sodium
not
carbonate
calculated,
base
it's
acts
really
as acid
calculated, like you get the
and
percent
acid.
for each thing, and then I weight each
phenols
category
and tannins. Tannins that phenols
based on that, so 60% of your grade comes from that, right? 15% of your grade comes from that. Download the final grade calculator, and see what it tells you.
Alright,
But it should be off the percentages.
okay, so just not, don't pay attention to the grade.
But it's giving you,
And caffeine flows the.
calculate it and see if you get a 77%.
I know they separate.
And you can send me an email that says, check the structure
But
and
that's all-
please double check the
all it was,
calculation on my grade, and I will
That
check.
they trap them. The base pulls the acid or something like that. I'll find out. Oh, this won't be on there. Did he all wanna, do me a favor? Okay.
You're good.
I was hoping.
Nick, you're gonna go on our solid-
Can y'all
I know,
call my
right?
phone? I don't know where it's at. I think someone might snag this for me.
You're what?
My phone. Why would it be?
Call it?
Yeah. Why would it be in here? No, it wouldn't be in here. I'm saying like whoever has it, hopefully it brings it to pocket. I don't know where it's
I
at.
think someone in this group, if that's not me,
No, bro, no, I'm saying if
not
someone
your
grabbed it for me, like, hey, let me grab this phone and left it. It'll ring. and
It
like,
went
yeah.
straight to voice.
I know you gotta call twice.
So do not disturb.
Yeah.
This is where it goes.
I hope someone snagged it for me.
What's lecture? Can you, let me know what's that one. Stop. The last slide. You stop.
I think it's legit, so. You know the number? Number 59. I don't know, today's gonna be.
59?
Yeah. Oh. Actually,
Oh,
I
how
got
many-
3 hours.
Oh, sorry. I was like, holy shit.
At 10:30, I woke up at 1:30.
Sorry, well.
No, an accident. That's that,
And you
that's,
sit up six?
yes, that's that work schedule right there.
You did a lot of.
That's that,
How's that,
that's
That
that,
was you that week, that, right?
yeah. I just didn't have the energy to come talk to you that morning.
I was kind of all the way down.
Catching up to me now, though.
Really?
I have no sleeping schedule. Like, it's just stupid.
I went to bed at 2, woke up at 6.
Went to bed at 10, woke up at 1:30. Now it's been up since. I'm an idiot.
Do you guys need a big stop clock, guys?
I would like.
I mean, do we have the whole class period?
Yeah, you had 2 hours, more than class, like, you know.
If I can't finish.
If you want more than 2 hours, that will be fine.